Mm. Hello everyone. My name is Roberto Blanco and my lab partner is Eros Aranda. Today we're over the Phage lab, basically just dilutions. This is lab report number 12. Okay, introduction. This experiment focused on determining the concentration or titer of a bacteriophage suspension under the highly sensitive plaque assay method. Bacteriophages are viruses that specifically infect bacteria and when plated with a suspected host, in this case, E. coli. In a solid medium, they form clear zones called plaques where the host bacteria have been lysed. To ensure an accurate count of these plaques, a zero dilution technique was first employed to systematically reduce the phage concentration of an individual, so as 10 to the power of -1, stock down to the 10 to the power of -6. The diluted phage samples were then mixed with the host, E. coli, and multi-top agar before beginning pour on the LB plates. The ultimate objective was to count the number of plaques forming units and countable plaques to calculate the original concentration of each stock. So basically, the materials today, or the day of the lab was 10 to the -1 dilutions of phages in macro centrifuge tube, 4 LB plates, 9 macro centrifuge tubes, still water, micropipettes and tips, and 4 large test tubes. The cultures, in all of them, we use E. coli. So basically, this lab, it was just dilutions, nothing crazy. It was little pipette, very easy. We use those pipettes with the correct measure. Of course, we had to change the pipette number and move from there. So procedure, like I was saying before, make a zero dilution of phages on still water. So we obtain a phage. This is a 10-1. The dilution from the lab tech that was obtained with the same procedure that you were about to perform. Obtain 5 of those tubes and label from 10 to -2, 10 to -3, 10 to -4, and so forth, all the way to 10 to -6. To find microcentrifuge tubes, sorry, add 900 microliters and still water to each one. From the 10 to -1 tube, obtain 100 microliters and add them to your 10 to -2 tube, and we mix it correctly. From the 10 to -2 tube, we obtain 100 again and add them to your negative to the power of 3 tube, and then we mix it correctly and repeat the steps until it reached -6. So it wasn't not even that hard. It was just like little mix. My lab partner did like by himself. Like it was just like help close it, open it, that's it. Like it wasn't a really hard procedure to do. Whole plate tighter from the zero dilution. Prepare a full LB plate by labeling the agar site. Date, type of phage from 1 or 2, diluted set. Obtain 4 of them, label them again E3, E4, E5, and E6. In this case, the E will stand for E. coli and the negative number will stand for the power of dilution. For example, E3 will be microcentrifuge 2 with E. coli and the phase of concentration to 10 to -3. So it wasn't that bad. From your original zero tubes, move 100 microliters from the dilution to the new E label tubes. So we use from those 100, we get 100 back. You now have 200 microliters in the tube, 100 from the phage and 100 from the E. coli. And we vortex the tubes briefly to mix. Allow the tube to sit on the bench for 10 minutes. So we just leave it there for actually 10 minutes. Make your way over the water bath and follow the items. 1 LB agar plate you label. The dilution microcentrifuge tube with E. coli and phage will label matches on the microcentrifuge tube. The follow steps must be done quickly and read carefully before you start. So basically, this one was the hard part because we obtained 4 large test tubes. We piped the phage with the bacteria, the 200 microliters from the microcentrifuge tube into the tube agar one at a time, and add 5 milliliters on multi-top agar from the water bath to a large test tube immediately. Pour the contents onto the respective LB plate. Stir gently 3 times to distribute the molten agar across the plate before it solidifies. Replace lid and allow to sit it under until the law sets 5 to 10 minutes. Keep the agar side on the table. You don't need to move a plate. Um, keep as label as possible. Invert it to incubate, paraffin, and place it to your basket and return it to incubate. So basically, in this one is just get it super fast after microwave, and after the microwave, just put it on the plate. It has to be super fast because if not, it's gonna get like cold down and it gets like hard. So the results in this case, again, we use E. coli in broth. So the first one, we can see a lot of those plaques that we said before. So every single thing over there, over here, like everything that's clear, that's the E. coli hanging there. So so over here we have a lot because it's 10 to negative 3. And every time like it was going down. So for example, in 10 to negative 4, we still see a lot, but like we can see some bubbles. That was a mistake that we did. Um like we still see a lot, but not like comparing to the first one. Um Again, it's because we start diluting with water. In the third one, okay, of course, like yeah, almost we don't see anything, maybe like 30 or 20 colonies. Like are these one small over here? Boom, boom, boom. And over here we make a mistake because we put in fast enough and also we got bubbles because we makes it too hard. Um, so that was a little mistake that me and my lab partner got. And number 10 to negative 6, we can only see like one, two, three, four colonies are those like clear areas. So basically, one was getting anything, and that's the purpose, right? To get less and less and less. Because imagine, like, if we get the same in all of them, that means we make a mistake. And in this case, we didn't. We was perfectly concluded. And conclusion, the successful exclusion of the zero dilutions on the plaque assay allowed for the accurate determination of the bacteriophage titer. By creating sequential to tenfold dilution, a range of concentration was plated, ensuring that at least one plate yield a statistically significant number of the plaque forming units. These results underscore the importance of precision and aseptic techniques and rapid plating to maintain the cell availability and prevent artifacts. The final tire conclusion provides the concentration of vital phages participant in the region stock, which is fundamental requirement of further research in phage biology and molecular genetics. And that's it. Thank you so much for watching. It was a pretty simple lab and almost the last one.